Okay, it's 3:05, so we're going to go ahead and get started. So welcome everyone. Thank you for joining us on Friday afternoon for our COVID-19 vaccine overview. This is something that the ID group, the ID Pharmacy group, wanted to put together just to kind of go over some of the vaccine data that has come out recently, and then also as an opportunity to discuss some concerns, some questions that have come up, and then to really fill in the gaps for understanding, as well as have an open discussion for potential questions that you may have after we discuss the data. So we're going to go ahead and jump in. The outline, so briefly we're going to talk about why this matters and kind of provide context of where we are in the pandemic and kind of specific data for UCLA currently. We're going to be discussing the currently authorized vaccines and their efficacy data and also safety results. And then Christine will be discussing some of the vaccine pipeline, things to be expecting in the coming months. Some information regarding special populations and also frequently asked questions. And then we'll also be providing a list of helpful resources for you all to refer back to, as well as have an opportunity for some open discussion, as I mentioned previously. So looking into why it matters, we're a year into the pandemic at this point, and it has become relatively easy to become desensitized to the numbers and kind of the inundation of data, of hospitalizations and deaths. But I did want to pause and take a step back and just assess where we are currently at UCLA, and then where we are currently as a nation in the pandemic. So these numbers are looking at deaths nationwide. As far as they were reported on December 30th. And on December 30th, there was about almost 4,000 deaths. And I think that one of the things that has become easy to kind of normalize is just these extremely high numbers of daily death tolls. But just to provide some context, 3,800 deaths is equivalent to 159 people dying every hour of this disease. And as far as numbers for UCLA, I previously updated this slide on the 4th of January, but each subsequent day we had an increasing number of inpatient admissions. So these numbers are actually updated as of yesterday. So looking on the right, the top right, we have 205 patients, inpatient visits that were encountered yesterday, split about evenly between Ronald Reagan and Santa Monica, 100 in each site. And then looking at the curve on this, the COVID-19 inpatient, this is our UC system, and this is over the past year. So just looking at this curve, we can kind of see that, back in the middle of the summer when we were really having a heavy burden of COVID-19 inpatients, that was our peak prior to this current surge. And we can just see that the current surge really dwarfs those numbers. And we're seeing numbers prior to this past couple of months that we had not ever seen previously in the pandemic. And this translates to 61 patients who are currently in our ICU, which takes up 47% of our capacity, as well as 40 patients currently on mechanical ventilation, and then 6 patients requiring ECMO. So this, once again, and no one really needs to be reminded of this, but this is a devastating disease and is occupying a large portion of our inpatient census as well as our ICUs. And one thing that I wanted to mention is that this is not simply a disease for the elderly and patients with comorbidities. There are a large proportion of patients nationally within the age range of working age patients that require hospitalization. In fact, this is national data, or these are national data that show about 47% of hospitalizations are patients of working age in the age range of 18 to 64. And if you look at our COVID inpatient service right now, at least the ID consult COVID patients, the median age currently on service is 45 years old, with an interquartile range of 40 to 62. So these are not all patients, that are elderly and frail. These are patients of working age and certainly younger. And then looking at at California data as far as deaths, and really speaking to the fact that this is not solely a disease that affects elderly patient populations, although they are certainly at highest risk. About 25% or a quarter of deaths are patients in working age in California as well. So that just provides a little bit of context in where we are currently in comparison to where we have come from this past year, and certainly in the midst of an incredible surge here at UCLA. So then looking at the currently authorized vaccines, of which there are two approved through the emergency use authorization pathway by the FDA. We have the Pfizer-BioNTech BNT162b2, and then the Moderna mRNA 1273, both of which are mRNA vaccine candidates or vaccines. So drilling into the Pfizer-BioNTech vaccine, which was the first to be approved on December 11th or authorized. It is a 2-dose series, 21 days apart. Logistically, there is a 4-day grace period for that second dose. It can be administered between 17 and 25 days after the first. However, if the second dose needs to be given late, this is permissible and it should be given as soon as possible. And there are no recommendations for repeating doses if there's a late dose. Notably, second doses should be of the same product. So if the patient received Pfizer for their first vaccine dose, they should receive Pfizer for their subsequent dose. And then also, so looking at the efficacy from the recently published large phase 3 interim data of the 37,000 patients who received both doses in the trial, the vaccine demonstrated a 95% efficacy rate in reducing symptomatic COVID infections. And then also for severe disease, there was a 75% reduction observed. And we see the numbers at the bottom here. So looking on the left, there's the number of symptomatic cases in the vaccine group, which was 8 versus 162 in the placebo arm. And then for severe COVID, it was 1 versus 9. So this is an objectively highly efficacious vaccine, as I've discussed with a few colleagues today, in comparison to our annual influenza vaccine, which we typically see a vaccine efficacy rate in the 60 percentile range in a good flu season. These are incredible efficacy numbers. And looking at the subgroups, so vaccine efficacy in the various subgroups. Can I ask everyone to mute, please, just so that we can all hear? So looking at the subgroups throughout the study, we can see that the efficacy held up very well in pretty much every subgroup that was assessed. Notably, these subgroups were not powered to significantly detect the primary efficacy endpoint. However, we can look at the point estimates, which are highlighted right here, and we see that nearly all of the vaccine efficacy point estimates are within the 90 to 100 percentile range, really holding around the primary efficacy endpoint about 95%. And I've kind of highlighted a few of the subgroups of interest for us. So first, patients who are at risk for severe disease, defined as patients with chronic lung disease, cardiac disease, severe obesity, diabetes, liver disease, and then also stable controlled HIV. We can see that the vaccine held up very well, 95% in this subgroup, which is incredible. And then also in our elderly patient population who are also at risk with an underlying comorbidity, we do see a good efficacy in this subgroup as well, which is notable because we typically see a reduced efficacy for our elderly patients because of waning immune system function and immune senescence in the elderly age. Also, notably, obese patients had comparable efficacy, and we certainly know that obese patients are at higher risk for severe disease. And the same is true for elderly patients who are obese as well. I did just want to point out, and this will be sent out in the slides that we provide, this summary video. It's a 3-minute video on the New England Journal of Medicine website. If you go to this link, it's just a really quick recap of the vaccine phase 3 trial design, safety and efficacy highlights. And it's really nice to look through or to watch as a refresher, as well as something that you can share with your colleagues if you ever get any questions. So moving on to the Moderna mRNA vaccine, which was approved via the emergency use authorization pathway as well. This is also a 2-dose series. It's 28 days apart instead of 21. Once again, there's a 4-day grace period for that second dose, with the same recommendations holding true. If the dose is late, give it as soon as possible without the need for any repeat doses. And also, it's important to maintain the same vaccine product. If you receive Moderna for the first, or if a patient received Moderna for their first dose, second dose should be Moderna as well. There is no recommendations for any catch-up or repeat doses if the second dose is inadvertently swapped. So if a patient received Pfizer and then inadvertently received Moderna for their second dose, there's no recommendations to repeat the series or do anything special. But it is recommended to just maintain the same vaccine product for both doses. Looking at efficacy in the 26,000 patients in the phase 3 trial interim data sets that received both doses, we see a comparable efficacy of a 94% reduction in symptomatic disease. And then also there were no patients in the vaccine arm that developed severe COVID-19, which is reassuring as well. We can see that the exact numbers here. So symptomatic COVID, 11 patients in the vaccine arm versus 185 in the placebo arm developed symptomatic disease, whereas 0 in the vaccine versus 30 in the placebo group developed severe COVID. So looking at the subgroups, once again, the efficacy held up well across all subgroups that were studied. Just highlighting a few here for you all. The patients in the 65 to 75 age range, there was a 100% vaccine efficacy in this subgroup. And then for the patients 75 and older, the same thing was seen there, 100% efficacy. Once again, also for our elderly patients who are at risk for severe COVID, we see 100% efficacy there. And also looking at different sexes, so male patients we know are also at high risk for severe disease, and we see comparable efficacy between the sexes. So all across the board, once again, we have an incredibly effective vaccine, and all subgroups seem to have comparable point estimates for their efficacy, but we do have to interpret these data cautiously as they were not powered to detect differences based on these subgroups alone. The same holds true for the Moderna study in the New England Journal of Medicine. If you go to this link, you can find a quick 3-minute summary video that you can watch yourself, you can share with your colleagues. All it requires is signing up for the New England Journal for an account with the New England Journal of Medicine, which is really quick. And these are really nice summary videos, and they're really quick to refresh your memory on the specific numbers. So one thing I did want to point out, these trials, the primary endpoints were assessing the impact of the vaccine on symptomatic COVID disease, which is not equivalent to COVID infection in general. So it is important that we all remain cognizant that it is possible for a vaccine to prevent symptomatic disease while not preventing asymptomatic infection. And because of this, we still do need to wear a mask, social distance, and wash our hands. And until we have further evidence to support or to refute the vaccine's ability to prevent infection and asymptomatic spread. So there are some preliminary data on asymptomatic infection prevention from the Moderna trial. In patients who were baseline seronegative in this study, who subsequently received their first dose and then before their second dose had a positive PCR result without symptoms, there was a 63% relative reduction in asymptomatic infection detected in the vaccine group. So we see this is overall very low numbers, but the asymptomatic infection rate dropped from 0.3% to 0.1% in the vaccine arm. These are just preliminary results right now. However, they are reassuring that there is potentially the benefit of preventing asymptomatic infection. However, we still don't have enough information to make any definitive recommendations based off of these data alone. And there are several limitations with this analysis, most notably of which it's unclear how soon after the vaccine that these infections occurred, or if patients subsequently developed symptoms later, and if these patients were simply pre-symptomatic and subsequently developed COVID disease. So a little bit too preliminary to make any recommendations, but these results are hopefully promising for the future of these vaccines. So looking at safety data, the most common adverse events in the Pfizer-BioNTech trial were injection site pain for the local adverse events, and then for systemic adverse events, fatigue, headache, muscle and joint pain, and then chills were the most commonly noted. Notably, the adverse events were more common in vaccine recipients who were receiving their second dose, and then also in patients who are in the younger subgroup, so less than 55 years of age. It is important to note that severe systemic events were relatively uncommon, so less than 2% of the vaccine recipients or of all patients in the trial, 1.1% in the vaccine group versus 0.6% in the placebo. There were 4 cases of Bell's palsy in the vaccine group, which is consistent with the background rate of the incidence of Bell's palsy. This is being assessed in an ongoing manner, and there are surveillance systems in place, just like with any other vaccine program, to continue to monitor for any signals. And then in the phase 4 post-marketing data, there were 6 cases of anaphylaxis reported in the first 272,000 doses administered. This equates to this is in contrast to the background rate of 1 to 2 anaphylaxis events per 1 million doses administered. I looked just before I got on here. There's actually been some updated data in this realm for anaphylaxis. There were 21 cases reported in the first 1.9 million doses administered, which it roughly translates to about 11.1 anaphylaxis events per 1 million doses. So this does appear to be higher than the standard background rate with historical vaccine data. And we'll talk a little bit about what the putative agent in the vaccine component that is suspected to be causing these allergic reactions in a few slides here. Notably, there were no increases in neurologic, neuroinflammatory, or thrombotic events detected in the trials in the vaccine arm. So that is comforting. And then looking at the subgroups broken down by dose 1 versus dose 2. On the left here, we have the younger cohort, 16 to 55 years of age, compared to the older cohort of 55 years or greater. And we see that the numbers are a little bit higher in the younger patients comparatively. And then we also see that the incidence of adverse events are also higher in the second dose compared to the first dose for systemic events. And we see that for both age cohorts. So looking at Moderna's vaccine, once again, the most common adverse events, injection site pain for local adverse events, and then for systemic, fatigue, headache, muscle pain, joint pain, and chills being the most commonly reported adverse events. These were more common following the second dose, and then also in the younger patients as well. And also for Moderna's vaccine, severe adverse events were exceedingly rare, 1.5% in the vaccine group versus 1.3% in placebo. In this trial, there were 3 cases of Bell's palsy in the vaccine group versus 1 in the placebo group, which is also consistent with the background rate. No significant signals of an imbalance between arms, and ongoing monitoring is being conducted. And in this trial, there were no anaphylaxis cases in the clinical trial, as well as no neurologic or thrombotic signals observed. So moving on to what could potentially explain the seemingly higher than standard rate of anaphylaxis. The mRNA vaccines are formulated with lipid nanoparticles in order to preserve the mRNA transcript from degradation, and also to adjust the hydrophilicity for transport. It is believed that the polyethylene glycol, which is the nanoparticle stabilizer, is potentially what could be causing these allergic events. Prior to these vaccine candidates, the polyethylene glycol stabilizers haven't been used widely in vaccine candidates that are approved on the market. So it is certainly something that allergy and immunology experts have suggested as the potential cause of these allergic events. The CDC has recommended that only persons with a known allergy to any of the components of the vaccine be excluded from vaccination. If you go to this reference right here, there is a list of all ingredients for all the vaccine candidates, both approved and on the horizon, the viral vector vaccines as well. Just something to, just for everyone's reference, the Pfizer vaccine has a polyethylene glycol 2000 components. The Moderna has one as well that's slightly modified. And then the AstraZeneca Oxford adenoviral vector vaccine has a derivative that's not a polyethylene glycol substrate, but it is something similar, the polysorbate 80. So the CDC recommends that if you have a history or a known allergy to a component of the vaccine, that you should omit or be excluded from vaccination. And if you have an allergic reaction to the first dose, it is recommended that you omit your second dose. Notably, polyethylene glycol is a compound used in other medications like contrast dyes, injectable methylprednisolone, and then also bowel preps. So if you have a history of allergic reaction to any of these, it is potentially worth discussing with an allergy expert to determine whether or not it is safe for you to proceed with the COVID vaccines. One last point before I turn it over to Christine. There has been some reports of allergic or adverse reactions in patients who have a history of dermal fillers, with swelling and inflammation reported around the dermal filler site. There has been a dermatologist that has weighed in on this phenomenon, and she stated that this should not stop patients from getting the vaccine, and that all dermal filler reactions were easily treated by medical personnel. However, if you do have an allergic reaction, or if a patient has an allergic reaction, the same recommendation holds true. The second dose should be omitted in that instance. Okay, and I will turn it over to Christine to talk about the pipeline and frequently asked questions.
Thank you, Matt. Can everyone hear me okay?
Yep.
So basically for the corona vaccines, there are 5 modalities that are trying to, of vaccines that are in process of being formulated. I'm going to go from the most traditional to the one from right to left, which is the mRNA being the most recent and most novel, potentially with the most questions. So starting from the right, we have the inactivated vaccine, which basically is whole virus grown with destroyed genetic material, and it's usually inactivated by chemicals or by heat, so that when it's injected into the body, it doesn't actually cause disease. The pros and cons in regards to that is that it has an immune response for a broad target range because you're actually injecting the entire virus. It is a long-standing proven technology, since the most familiar that we are familiar with is the quadrivalent annual influenza vaccine that we get annually as healthcare workers. It is time consuming to grow depending on the pathogen. For example, like I want to say half of our flu vaccines are grown in chicken eggs, so that does take a while to replicate in order for that to occur. And specific facilities have to be assigned in order to produce the vaccines for this, because they're handling whole virus, so they have to be careful in regards to strict precautions. The primary pharmaceutical company that actually has targeted the inactivated vaccine modality for COVID is Sinovac, which is coming based from China, and that is they've already sold, I want to say millions of doses already globally, and already had administered several of their own patients in their own country, and that's called the CoronaVac. If you might have heard it in the news recently, China has also created their own vaccine, but it's in the inactivated vaccine modality. And then moving on to the live attenuated vaccinations, that basically is also whole virus, but it's been weakened, and it could replicate, but not to the extent that it can cause disease. And again, it gives a strong immune response. It can be given intranasally, which is what we have, for example, flu block for the influenza. It's familiar, long-standing technology that we are familiar with. And then but then the downside is that it's time-consuming to grow. You still need specific facilities for production because you're handling whole viruses again, and then there's safety concerns for those facilities. And then there's two companies in the pipeline who are trying to work on the COVID vaccine using the live attenuated modality. Moving on to the next protein subunit modality. So this basically is fragments of the virus where they are expressed through various cell lines, and then instead of you're not handling the whole virus, so it does not cause disease. However, they're less likely to be recognized by your immune system, so you may have a weaker immune response, and then so they're often combined with chemical adjuvants to actually stimulate a stronger immune response. All subunit vaccines are usually made using living organisms, such as bacteria or yeast, to grow them. And so strict hygiene is required to avoid contamination. Some examples that we've had in the past is hepatitis B, A cellular pertussis. So in general, for the corona, there's two pharmaceutical companies that are trying to formulate a coronavirus vaccine using this modality, but the spike protein is hard to create because you're using a fragment of the antigen and still trying to create an immune response. So they'll probably have to combine it with the chemical adjuvant to elicit a stronger immune response. Moving on to the next modality, the viral vector. So there are two types of viral vector-based vaccines. There's non-replicating and there's replicating. For the coronavirus vaccines, they're all trying to do the non-replicating viral vector modality. And so basically the vaccine mimics similarly to what you do in natural infection it's based on another virus. You have a shell of a non-replicating virus, usually adenovirus, was the most common one because it binds to your upper respiratory tract really well. And then inside the viral vector is the genetic code to actually make the antigen. And so it basically tells your cells to create antigen so that it creates the immune response. So this is also considered relatively novel technology. The first licensed vaccine that we have using this modality is the Ebola virus vaccine. And it provides good immune response. The downside to this modality is that because it has to be an adenovirus that is not recognized by most of the human population, so that it won't be neutralized before it goes into the cell to produce the antigen. So I think some of the viral vectors that the coronavirus vaccines are trying to look at is chimpanzee adenovirus as the viral vector, so that the body doesn't neutralize it beforehand. So you're actually having to look for viral vectors that are not recognized by most of the most humans across globally, because the body might be able to neutralize it. So that is a downside. It makes it harder to produce in certain ways. There are 3 of our pharmaceutical companies that are trying to address this modality, AstraZeneca being one of them, Johnson & Johnson is the other one that is using this modality for the coronavirus vaccine. And then most lastly, the ones that are EUA approved is the mRNA vaccines. And this basically is, they're just mRNA or nucleic acid vaccines are basically a small genetic code that creates that basically tells the cells to create the antigen to mount an immune response. And so you're not actually giving any part of the virus in, it's just genetic code. So it should not cause disease. And then once a pathogen's genome has been sequenced, it's relatively quick and easy to design a vaccine against this protein. So for example, Moderna's RNA vaccine against COVID, once they entered the clinical trials, they were able to have, within 2 months of the genome of SARS-CoV-2 being sequenced, they were able to actually make the vaccine. So in regards to the advantage of this modality, it's very easy and quick to design, large scale production. It's relatively new technology. You do have to worry about the stability issues of handling nucleic acid. So that is potentially why there's issues with storage with, like, for example, this Pfizer. And so the 2 pharmaceutical companies that are EUA approved is Moderna and the Pfizer, which we're all familiar with. Matt, can I have the next slide, please? So just in regards to what's coming up next, the key vaccines that are coming up next with ongoing phase 3 studies is AstraZeneca and Johnson & Johnson's, which is the viral vector modality. And preliminarily from the phase 3 studies, the AstraZeneca, which is 2 doses, is showing to be 70.4% efficacy. I think they're still trying to figure out the dosing regimen with the low dose versus standard dose in regards to efficacy. And then for Johnson & Johnson's, it's just 1 dose, and the phase 3 results are expected sometime maybe this month or next month. And then of course there's the protein, and there's another modality that's also in phase 3 that's expecting to come in early 2021 of this year. So that's Novavax, and that's 2 doses, and that's using the protein-based modality. So just as a timeline, one of our colleagues created a timeline of basically how many phase 1 trials had applied in regards to what type of modalities for coronavirus vaccines had created, but then it became to just being 3 being approved for use, 2 of them in the United States, 1 of them being in China, and then 1 was abandoned. So I mean, there's been a lot of applications, and thus far this is what we have so far. Next slide, Matt. Thank you. So this is just a common question because of the way that the speed of the vaccine had been created. There's been presumptions that it was taken or manufactured in regards to this technology in a very quick turnaround time. But in fact, this mRNA vaccine technology actually has been studied since 1993. And then from the article that I had extracted, basically this is a timeline from the 1990s to current time, where they've actually tried to perfect the development of this technology. Most of the development restrictions came from the fact that we were dealing with nucleic acid instability and potential degradation in the body, and also handling it on the outside exogenously. And then there's high innate immunogenicity, so they had to figure out how to tweak the the nucleic acids on the mRNA in order to have the appropriate immunogenicity in the body, and then also what to attach it to in order to stabilize it for it to get to the cells correctly. So this has been actually a long process that has already been started from a while back. And then in the past, I want to say since 2017, they have had they've had several vaccines already looking at other viruses before the coronavirus. They were looking at influenza, Zika, rabies, and CMV, and they're all in phase 1 and 2 studies that have been started in the past 3-4 years that have been utilizing this modality as well. And I think the reason why it hasn't actually gone more progressive in terms of the technology is because it hasn't been well funded. There isn't a sense of urgency like the coronavirus because of the pandemic. Next slide, Matt. Just to speed ahead, basically the 4 factors that allowed for the process to speed up development was the fact that there was the Operation Warp Speed that allowed for $10 billion of funding. And then so they also were able to do combined phase 1, 2, or 2 and 3 in tandem concurrently versus before it used to be consecutive versus concurrently. And then because of the number of cases that we have, there's been rapid enrollment and recruitment. And then so all in all, also because it doesn't have to be grown or you don't have to handle the actual virus in special facilities, the mRNA vaccine production is also made really fast, like within a few weeks time versus, for example, the influenza vaccine takes up to like 6 months to manufacture, for example. And then otherwise, I mean, the requirements by the FDA that were followed for this approval were still followed very stringently. And then all the documents, of course, are public. They're reviewed by independent committees with the vaccine-related biological products advisory committee and also the advisory committee for immunization practices. So in regards to the long-term side effects that have been concerning for the 2 mRNA vaccines, the side effects have been monitoring for up to 4 months. They've been having ongoing post-marketing safety checks, and then have made the safety check to report vaccine adverse events through a mobile app, which Matt and I will look into or explain further. And then so majority of the adverse events for majority of vaccines only occur within the first several months of the vaccine. And then otherwise, even like for example, GBS, I believe the post-marketing surveillance for some of the other vaccines for safety was actually within the first few months of post-vaccine injection also. So, I mean, in regards to safety issues, it's really, really low. And the fact that we have such high numbers is something to actually take into account as well. Next slide, please. And then these are just special populations. Matt and I have been receiving several questions from different individuals regarding whether or not they should be vaccinated based on their past medical history or their current situation. This is just a summary of patients with a history of a past SARS-CoV-2 infection. You should still vaccinate regardless of prior infection. There is some CDC guidance that says you may be able to defer 3 months post your infection because your antibodies might still be around. But regardless though, you should still consider vaccination. And then for persons with known current SARS-CoV-2 infection, you would delay the vaccine until recovery, until when your isolation is completed. I received a question about that yesterday. There was a patient who had an active SARS-CoV-2 infection. They were eligible to get their vaccine. They were a healthcare worker, but I advised them to actually stay home until their sickness was completed and their isolation was done. And then patients who have previously received passive antibody therapy, for example, like convalescent plasma, any of the monoclonal antibodies for COVID. There isn't really any data, but CDC recommends that you defer vaccination for at least 3 months or 90 days to avoid the efficacy, to avoid diminishing the vaccine efficacy. And then for immunocompromised persons, those patients were excluded from the trials. There isn't really any data, but most organizations are recommending that unless contraindicated, you should get the vaccine as well. For pregnant or breastfeeding persons, I'm going to probably go a little bit more in detail with that on the next slide, because I've received actually a lot of questions regarding this. But for the most part, Fetal Medicine Society has recommended the vaccine to be available to all pregnant or lactating women. It's usually in conjunction with a discussion with your physician. And then public health recommendations is that protection is not immediate. It usually, even for any regular vaccine, it usually takes 2 weeks to get full immunity. And this is 2 doses, so you might not even get full immunity till like 2 months post the second dose, or 2 weeks post the second dose, excuse me. And then you continue to follow current distancing and protection guidance. And then patients with anaphylaxis to injectable therapies, Matt had already talked about this. The primary concern is related to the pegylated compound. Next slide. So just to go a little bit more into the pregnancy lactation, for those patients, there isn't any data. However, during the clinical trials, in the Moderna trial, they excluded pregnant patients, but then there were 13 patients that became pregnant on the trial. And then in Pfizer, the clinical trials, there were 23 patients that became pregnant while on, and there were no harm signals that has been made from those trials. And then the DART studies, which is basically animal reproductive toxicity studies, for both vaccines, Pfizer, the results are expected to come any day now. For the Moderna, there was no observed adverse events at the 100 micrograms level when assessing for pre and postnatal developmental effects. And then these are just the 3 societies regarding on their stance on vaccinations for pregnant and lactating patients. And basically for the ACOG, they say that pregnant patients should make their own decision, but it shouldn't be discouraged, basically. And then lactating individuals should be offered the vaccine once it's eligible. And then for the Fetal Medicine Society recommendations, they strongly recommend these patients to actually have access to the vaccine. I've had 2 COVID ID attendings who are both who both are pregnant, and and based on their discussion with their fetal medicine doctor, they've already received their COVID vaccine. And then for the CDC, they also say they should be eligible to get vaccinated for both patient populations and not be discouraged from taking it. Next slide, please. And then this is for the American Society of Transplantation. In regards to immunocompromised patients, there is no data. However, their stance is that all transplant candidates and their household members should be vaccinated when it becomes available. Vaccines are usually recommended greater than 2 weeks before the transplant, or starting at 1 to 6 months after the transplant. So that's the stance, even though there isn't any data to support that. But I believe because of the prevalence of and the severity and acuity of the COVID infection, they recommend that this vulnerable population get vaccinated. Next slide. And this is Matt's registration with the VSafe adverse effect monitoring on the smartphone. So if you actually turn on your camera and put it on this code and send in your contact information, they will check in with you regarding how are you feeling post vaccine dose administration. And then that's where you may be able to submit your side effects if there are any severe. So they are trying to capture as much safety data as much as possible for the general population, including and with our prevalence of smartphones, that's a good way to do it. So if you have any questions, I tried to do it and it didn't really go through for me. I didn't register through my phone, but I tried to submit the form. Matt successfully did it. So if you have any questions, please forward the questions regarding if you want to register for this check-in application with Matt. And then some questions that have come up also recently. Again, what do I do if I previously had COVID-19? You should get it irrespective of the fact that you had a previous infection. If you have a current infection, again, you should wait until you isolate and recover. And then usually the 10 days post, if you're outpatient. And then if you previously had COVID-19, will I have worse side effects from the vaccine? That was a question that came up about a week ago. It's not entirely clear, but patients who had seropositive versus seronegative at baseline, patients who had no prior infection previously, it appears that they might have a slightly higher incidence of adverse events than patients who are seropositive or who had previously had a past COVID infection, but it's primarily very small numbers, and it primarily shows not an increased risk for side effects if you had COVID-19 in the past. So at UCLA, there was a question whether or not when will it roll out to ambulatory patients, since essential workers and healthcare workers have predominantly been vaccinated. So infection control has told me that ambulatory patients will potentially have an announcement by the end of this month to have an invite to get vaccinated for ambulatory care patients, most likely through their MyChart UCLA. That was a question yesterday that was sent. So in case you are if any of your ambulatory or UCLA healthcare patients ask, it's most likely the invitation is gonna be sent out later this month. And then next slide. These are just a list of resources that we have listed in regards to if you want to know more information about the vaccines. These are really good that can explain in further detail, and also the 3-minute videos that Matt had mentioned earlier in the 2 other slides. I think we can open the floor for any questions.
Yeah, mute. Thanks, Christine. That was a great summary. Does anyone have any questions that were not addressed in the presentation? Any things that's top of mind for you all? I have one in the chat. Okay, so we have a question from Linda. Does the vaccine affect COVID testing later? That's an excellent question. It would depend on the modality of the testing. Certainly, if it was an antibody titer test, qualitative test, you would see a positive result because of the immunity from the vaccine. However, it shouldn't affect a PCR nucleic acid-based test. Okay, looks like Brandon has a question on the EUA antibody test and its availability. Brandon, I do not know the answer to that. Christine, Meg, Ross, anyone have an answer?
I'm not aware of any. We can probably look into it to see if there's any data available to see the timeline, but I actually haven't heard anything about it.
Yeah, we can certainly follow up with you, Brandon. Great question.
And then there's Chris's question. Did you see that?
I did not.
Um,
Oh, yes. Okay. So is there any updated data on the durability of vaccine protection? That's a very timely question, Chris. There was a publication recently with a kind of a follow-up subgroup from the Moderna trial. was about 117 patients, or 120 patients roughly, and they followed them up about 3 months post-second dose. And they saw that although there was a small waning in the antibody, the neutralizing antibody titers over time, they still were, there was still a durable residual geometric mean titer of the neutralizing antibody, and it was also higher than the banked convalescent plasma for patients who were median 30 days from the onset of illness. So even though there was a slight weaning of the antibody response over 3 months post second dose, the titers were still higher than titers on average for patients that we've salvaged convalescent plasma from. So it seems like at least until 3 months out, we're still seeing really durable responses. Now that is something that needs to be considered with that is we don't have a correlate for protection. We don't know necessarily what the magic number for a neutralizing antibody titer is. And so we see kind of this response persisting for some time, but we don't fully understand how to interpret that. But it is promising to see the antibody titers still being high that far out from the vaccine. Okay, well, thank you all for your attention. I think that we might not have any more questions. These slides will be sent out as an email. This recording will be posted. And then certainly we're all available for any questions that you have, after this, as you begin to think of more, or as you get questions from patients or colleagues. So please do not hesitate to reach out to any one of us with any questions that you may have, and have a great weekend.
I was expecting.